Hello everyone, my name is Nicolás Zúñiga, and today I will present my first talk called Vesicular glutamate transporter genes, origin, evolution, and molecular signatures underpinning glutamate transport in animals. As an introduction, cell-to-cell communication corresponds to the transfer of information either between neighboring or distantly related cells. In the case of distant cell-to-cell communication, the sending of information is conducted by means of an specialized system that involves the process of a synapse, where a neuron passes the information, electrical or chemical, through an action potential to another neuron. One of the most relevant synaptic types occurring in the nervous system in animals is the excitatory synapse, where neurotransmitters such as glutamate are released into synaptic cleft and activate receptors that open ion channels in the postsynaptic neuron. In chordates, vesicular glutamate transporters, VGLUTs, are transmembrane transporters that increase the quantum size within the synaptic vesicle, loading glutamate vesicles by binding to specific residues. VGLUTs are regulated by chloride and proton binding residues, as well as by endocytic tyrosine-like and polyproline domains that bind to adaptor proteins and endophilin, respectively. The activation of glutamate receptor has been detected in a wide variety of species, even though those that lack a condensed or centralized nervous system, such as cnidarians. In spite of this, the origin and evolution of VGLUT functionality throughout the history of animals remain poorly understood. In an in-depth analysis comprising 110 eukaryotic genomes, we were able to detect the 9 members of the VGLUT family, SLC17. We detect that the transport of anions in this family corresponds to its central function, with sequential losses in the stem leading to fungi and ctenophora. The vesicular transport of nucleotide and sialic acid were detected in the last common ancestor of Guanozoa, while the emergence of glutamate transporter that not use vesicles was present in the last common ancestor of eumetazoa, whereas the GLUT transporter that use vesicles appear in the last common ancestor of Nephrozoa. By analyzing the evolution of glutamate transporter more deeply, we detect a clear separation of this transporter in 3 groups. First, we detect a group of glutamate transporter that are not vesicular, comprising different species like Arthropoda, Cnidaria, and Mollusca, whereas there is a second group of glutamate transporter, which are vesicular and is also distributed in different non-chordate species. Both GLUTs and VGLUTs are characterized by lineage-specific expansion and differential gene losses over animal evolution. Finally, there is a third group that is specific to chordates and contains the 3 well-known members of vesicular glutamate transporters, I mean VGLUT1, 2, and 3. To further understand glutamate transport functionalities, we conducted a structural analysis focusing on already known functional motifs and domains. From this analysis, we observed that glutamate binding and regulation through chloride and protons are present in most modern animals, except for nematodes. On the other hand, the emergence of endocytic regulation was detected on the dawn of bilateria, with sequential losses in hemichordates, annelid, rotifera, and nematodes. Additionally, polyprotein motifs were only observed in chordates, specifically in mammals, which may have a direct correlation with a more condensed or centralized nervous system. We observed that the fine-tuning co-regulation of glutamate transport correlates with nervous system condensation in animals. We propose that the complexity and condensation of nervous system observed in modern animals might be correlated with a stepwise acquisition of different modified domains, which are involved directly in glutamatergic signaling.